share my topic and my research, or the expert in this agriculture field. So this is a long title, but I make it shorter. It's MS. So let's talk about the modern technology and use for the rapid response of the agriculture-related biosafety issues. So okay, actually we already introduced myself. I don't have to mention again. Here is about my working place before I start my topic. We are in Beijing now, and my place is from further north of China, and it's almost the same latitude for Hawaii, United Hawaii. And those are the three places my career was spending on, so yeah, that's all. And those is the interesting plans we are doing, but the most important is we use this case to study why this biosafety issue is very important in my place, Hainan. This is the Hainan Island. Because it's a tropical area, in the winter time, mainland China, all the scientists will try to do the winter breeding from September and to the next year, May, so they go there for the breeding. They bring a lot of material. There's a lot of biosafety issues concerned. More than 500 institutes and more than 5,000 researchers was gathered there to do the research, and including the rice, corn, soybean, and so much. Not even mentioned some transgenic or some GMO plant was studied there. So there's a lot of concern about the biosafety, including the unintended insects disease, pathogen, quarantine insects disease, and pathogen and GMO material. So our group was focused on the biosafety regarding this, especially in our location, Hainan, during the winter breeding season. And my specific will focus on those issues bringing for the soil microorganism. Okay, so this topic is biosafety. Actually, there's two parts. One is the technology. It's called mass spectrometry-based microorganism identification technology, and the next one is rapid response. So in case if we can identify microorganisms by such like a barcode, so we can easily to scan the microorganism and you can get the result. So that will be considered as a rapid response. Okay, we know we can study the creature or the biology through species, genus, family, order, and all those ones. But how about those microorganisms? There have to be some way to do, but tell you, there's spend a lot of time consuming and labor consuming. For example, you use bacterial morphology or biochemical biophysiology or bacteriophage or even genome polymorphism. You can distinguish like monthly, even longer. Recently, we also have some other considered modern technology like a conserved gene sequencing or the whole genome sequencing can be okay to identify the microorganism, but it's still not considered rapid. Today I'm going to talk about we're going to use some other kind of idea to show how rapid to do the identification. For example, conserved gene sequencing, maybe we will know, is most people doing is about the 16S rDNA sequencing. That's the most conserved gene to do the classification and taxonomy. Let's go back to the ribosome protein, which is also considered 16R RNA. That's only one RNA, but generally in the life or in a bacteria or in a cell, there are more than 2 RNAs and more than 50 proteins. If we can gather the information in one time, it must be more accuracy and higher resolution than only one 16 rDNA sequence. So, and with development, this machine is called Matrix Assisted Laser Disposition and Ionization, MADI, can be used to analyze all the ribosome protein from a cell at once. And this is the E. coli DH5 alpha we can just one laser shoot, you can got all the ribosome protein. We can use all the information to identify these strains in a second. And since there's some example, like you can just acquire all the data from the one simple sample, and you can go to like something like a barcode, and this is specific to your sample, so you can just like barcoding your bacteria. Okay, this once we got this this kind of technology, we want to know their hair tolerance. What mean tolerance? Like what kind of is those culture or the media was affected this your resolution, or the culture time, or the storage condition, or the duration. Like you purify your bacteria in Hainan, you want send to USA for identification. Is that okay? So we try to optimize all the parameter. Like these strains we use from 6 hour cultivation to always 30 hour for the E. coli, and also we use different temperature to store the culture from 4 degree to room temperature from one day to about 2 weeks. So it's all result promise us a good resolution and considered to be high tolerance during the different transfer from the store from the lab to the different country for the identification. And also, we want to show the higher accuracy. We use about 79 isolates of Rhizobia strains to demonstrate how accuracy of this technology to compare with 16 RNA sequencing. From the left, you can see this is the dendrogram from the mass spec based identification technology, and on the right is the 16 RNA sequence. It's higher resolution and more accuracy. So to apply this, we develop a two-step identification. First, if it's unknown spectrum of the bacterial protein profile, we first was compared to determine which genus at the genus level. First we can use on the left we use the genus level. We know as they have a log score was much higher than other. Like for example, our blind sample was Rhizobia. It's not a branded Rhizobia or any other Rhizobia. It's just Rhizobia. And then next step, we just focus on all the species inside the Rhizobia, like all on the right side, and do the second identification. And we mark this as Rhizobia multi-hostium. It's like this one. So overall, it's just take 1 hour from isolation to get the result. This is this is just technology part. I will use my case study to see how this work is gonna be. It's about the GMO rice as for the endobacteria in genome rice. Recent 2 decades, more and more GMO crop was being introduced and cultivated. Meanwhile, like scientists and consumer will pay more attention to the environmental safety concerns. Our research was focused on endophytes. Very important was for century during the evolution to see they have a relationship with plants. So, but if we have introduce some other gene into this one, maybe we can find some effect about this one. This is already the idea, and then we use the rapid identification technology to screen the GMO rice. And we compare the endobacteria from GMO rice, which is transformed the BT gene, and as well parental cultivar, and we screened the whole growth stage, including seedling, tearing, flowering, and the fruiting stage. So in total, we isolate about 1,200 isolates from all the growth stage of two rice plant. And if you see, that is 664 from GMO rice and 529 from the wild type rice. And we use the rapid identification method, also validate with the 16 RNA sequence in the endobacterial isolate from the GMO rice was belong to 4 film, 16 class, 14 genus, and 92 species. And the endobacterial isolate from the wild type rice was belong to 50 genus and 99 species. And this was continued for the dominance of the endobacteria in the GMO rice. The dominance in the seedling, tearing, flowering, and the fruiting stage was different in GMO rice, but in the non-GMO, you can see all of them are Bacohoda species. Genus. This is what we found, and we also found some specific endobacteria in the GMO rice. There are more than 8 different categories, if you can see. On this column, this is GMO rice, and this is non-GMO rice. You can see this is only detected in the in GMO rice, only found in the seedling stage, tearing stage, flowering stage, and the fruit stage. This is only found in the non-GMO plant from the seedling, tearing, flowering, and the fruiting stage. And also we found some like this claw as only in GMO rice. So this is not the end of the story yet, because we need to confirm those bacteria. Maybe they can be an indicator of sensor to censor this change from GMO rice. I'm not going to show you further about that, but I will see. My next step is during this whole data collection, we're able to set up our in-house database, protein database, including those species. And if you have a sample which is maybe involved this species, we can identify it in 1 hour. And we also set up a database server, like the clients will submit their, no matter what kind of machine they get their data, they send the server, we can find in our database and give them the identification result. And this is the overall pipeline for service to do the identification service, like from the pure culture of environmental sample, you just shooting with the laser and get MS data. You can just search our database. If the log score was above 1.7, then it's pound. You got your result already. If it's less, we can use another level of protein preparation. This from the first step is only 15 minutes. If you use standard preparation, it's 30 minutes. If you use some other delicate protein preparation, it's about 1 hour. So in case none of those the log score was above 1.7, there must be a new species or something not involved in our database. We also can artificially learn this species and put in our database. So gradually, the database will be bigger and bigger, more valuable for the service. This is all the story I'm talking about. Thank you for the ITBB and HARG and UH. This is place I'm playing. Thank you.
good. And you say just for 1 hour, you have your results. And looks to have a lot of importance when it comes to classification of organisms and whatever. But my thinking is, how cost effective is it in terms of developing countries, which may not have state-of-art laboratories you have here? Is it user-friendly in terms of cost for low-developing countries? Thank you.
Okay, actually, this you're talking about the cost. This is quite. We have been tested this technology on different machine. It's machine independent. So any kind of machine from different country can do this one. And like I said, this is quite tolerance. So you can just isolate the bacteria or the microorganism from your sample and send to some facility to do the the mass spectrum, and you send to the database and got the result. It's for only in terms of the identification from the mass spectrum is quite low. I'm concerned about $5 to $20 per sample. That's considered including all the machine part of consuming. But if only because the time, if your sample is already already in our database, it only take 15 minutes to get the result. Yeah, that's quite applicable.
I have a question. It's very nice and very impressive. The time-consuming part, the limiting step, and I think also where you might lose something is the purification part, right? You have to isolate these strains before you can barcode them in no time. But this is the limiting factor, or?
Thank you. You actually raised a good question, but I'm not mentioning deep about this principle of this identification technology, because in the cell or any cell, the most richness protein is ribosome protein. It's considered more than 40% of the whole cell content. So it's just break the cell. It's quite easy to extract the DNA. It's about less than 15 minutes to extract the protein. It's not considered all the protein. It's only the richness protein.
But you have to isolate the strains before you do this, right? And this is the time. And also that when you're talking about endophytes, there may be some which you just can't isolate, so you will never learn about them. Is it right?
Yeah, that's true. That's the isolation part. Still labor consuming. But there do some researcher was try to extend this technology to use for example, people working on the nodule rhizobia who was nodulate on the legume tree, they can identify directly from the nodule, because nodule consider have a high concentration for rhizobia, which can directly identify. You just cut the nodule and smear this on the target and shooting with laser, you can get the result. But some other endobacteria, is the low concentration in the plant. That will be hard for you. We try on some virus in papaya. There was a ring spot virus who was considered higher concentration in the plant, but no, there's too many background. We still needed the isolation.
Do you have a good explanation for the differences you saw in this BT rice versus the control rice and endophytes? Because it doesn't make much sense that they are very different if it's a BT gene, but maybe there is a reason. Do you understand it?
This is kind of a more controversial topic about this. But since we have some gene was transformed to the species, there must be some change about metabolism, which may be during the centuries of evolution, the bacteria and the plant have a symbiosis relationship there. If you change the nutrition or any metabolism, that may affect the bacteria. But so far we didn't have the conclude yet.
But you are confident that these differences are very significant, right?
We need to further validate those to be indicative in reporter or sensor to be like this GMO non-GMO indicator. But so far we have more confident is we got those isolates. Now we need to bring back to the plant. It's really can be considered as an indicator or not.
Very interesting technology, but what I would like to ask is that because you are dealing with bacteria, how about, like, say I have a lot of rice accession, can I use to identify them?
Rice
Accession. Let's say I have
rice variety.
Rice variety, I have a lot of them, like maybe 100 or maybe up to 500. Can I use that technology?
Yeah, definitely you can do that. We have been working on ongoing subject was compare different of rice, including wild species rice, was try to not only the endobacteria but also the soil microorganism. Because we already, if you're talking about the rice bacteria, we already have a database. This is we have the literary research. This is considered most of the endobacteria already. So maybe it's more, you can expect them faster than other bacteria to be identified.